Am I audible? Am I audible to you? Good evening, everyone. Am I audible to you? You people are not coming to- Am I audible? Please tell me in the comment section. Am I audible to you? Good evening. So today we are going to discuss the topic that is your isolation of the DNA from animal blood. Sorry, human blood, not animal blood, human blood. Isolation of DNA from human blood. Okay, today the sample is what? Today we have a sample that is your human blood. And with the help of this human blood, we have to isolate the DNA. Okay. For the isolation of the DNA, we require a reagent. So first of all, we have to discuss the reagent which is used for the isolation technique. So the first reagent that we require is a lysis buffer. First reagent that we require is a lysis buffer. So this lysis buffer are prepared by the composition of different chemicals, and we have to prepare this lysis buffer around 100 ml. And the final pH of the lysis buffer will be 7.5. Okay, do you know how to measure that pH that I already told you in the yesterday class? So this lysis buffer are prepared by the composition of sucrose, which is required 10 points. Yes, Varun, please ask. 1.96 grams Triton X100, which is required 1 ml. Then the required magnesium chloride 100 milligrams, and at last we required fresh base. Sun is used in a plant tissue culture. Okay. This what will I give you? I will give you an answer. Explant is what? Explant is used in a plant tissue culture for the preparation of the plant in a laboratory. Preparation of the plant, preparation of the plant in a laboratory under the sterile conditions. For that, when we have to prepare a whole plant in a lab, okay, so we can take our X plant as the initiation. वही बताइए. हम्म. These plants are used for the production of the new plant in laboratory conditions. Okay? Clear, है? This question is not related to your syllabus, okay, because we are studying about the isolation of the DNA from the human blood. Okay, if you want to ask this, the question which is beyond from your syllabus, then you have to ask at the end of the section. Clear, Varun? So this base is required around 150 milligrams. Now, what we have to do, we dissolve all these chemicals into the 15 ml of distilled water. When it is completely dissolved, then we maintain its pH, we maintain its pH. After maintaining its pH, that is around 7.5, we filter it with the help of the filter paper. After filtration, we no, not filter. We maintain its pH, then we make its volume 15 ml. After making the volume 15 ml, we filter it, and after filtration, we store it at 4 degrees Celsius. Clear? This is how we prepare the lysis buffer. It is clear? Preparation of the lysis buffer is clear? Then next is your protease buffer. Protease buffer will be prepared for 50 ml, and its pH will be around 8. Okay. To get the preparation of the this protease buffer, we require two chemicals. The first is your sodium chloride, and the second one is your EDTA. Sodium chloride will be required around 128 grams for this 50 ml, and EDTA will be required around 2.44 grams for the 50 ml. Now, what we have to do, we dissolve all these chemicals that is your sodium chloride and EDTA into the 25 ml of the distilled water. Then we maintain its pH is that we make it swallow 15 ml with the help of distilled water. Then after that, we filter it and store it at room temperature. This is how we prepare the protein breakfast. Next something is your 5 molar of sodium chloride, which is prepared for the 50 ml. Again, with the help of this formula, that is weight is equal to molecular weight multiplied by molarity multiplied by volume given divided by 1000. Next chemical that we required is a 10% of SDX. This 10% of the LBS is again just prepared for the 25 ml. So we have to prepare like this 10 by 100 multiplied by 25. Then we have to prepare a last chemical that is your phenol chloroform solution. Phenol and chloroform solution. This is prepared in the ratio of 4 to 1, which means that if we take 4 ml of the phenol, then we take a 1 ml of the chloroform. Clear? Preparation of the reagent is clear. Preparation of the reagent is clear. Then after preparation of the chemicals, we have to record, we are going to discuss about the role of the chemicals. So the first role of the chemicals. The reagent. So the first chemical is your SDS and protease buffer. This SDS and protease buffer is required for the destruction of the cell and nucleus membrane. Next is your lysis buffer. It is used to remove the cell membrane. Then NaCl. NaCl is used as specific due to this NaCl specific salt concentration occurs which generate the detergent form due to which specific salt concentration occurs due to which detergent form and complex with nucleic acid. Then next chemical which is used as base, which is used to maintain the pH. Okay. We are discussing about the role of the chemical. So the first chemical that is used aqueous and protease buffer, which is used for the destruction of the cell and nuclear membrane. Then we require a lysis buffer for the like it is used to remove the cell membrane. Then NaCl is used in a space. This NaCl gives a specific salt concentration and due to which detergent form an insoluble complex with that nucleic acid. Then we require a stress base which is used to maintain the constant pH. Then EDTA is used as a chelating agent. To chelate the magnesium ion, which is essential for the DNA enzyme, which is essential factor for the DNA enzyme. This is the role of the chemical. Now come to the procedure part. Preparation of the chemical, role of the reagents is clear. Procedure. Then we require 0.5 ml of blood in which we will add 0.5 ml of the lysis buffer. Then we mix it properly by inverting it for the 1 to 2 minutes. Then after that we perform our centrifuge at 11,500 RPM for 5 minutes. Then we get what? We get the supernet and the palette. We discard that supernet and in a palette we will add again 400 microliter lysis buffer. And again we mix it properly and perform our centrifuge at 11,500 RPM for 5 minutes. And we will get what? We will get a supernet and the palette. And we discard that supernet. Now we have to repeat this step. Like again we will get the palette. Again we added 400 microliter of the lysis buffer, mix it properly. And again perform a centrifuge at 11,500 RPM for 5 minutes until we get a clear white color palette. Repeat this step until we get clear white palette. When we get the clear white palette, we of this app, in parallel, we are going to add our 100 microliter double distilled water. 400 microliter double distilled water. And we mix the pallet into that water. Mix the pallet to dissolve into the double distilled water. Then after that, we have to perform a centrifuge at 11,500 RPM for 5.5 minutes. Then we get what? Same. We get a supernatant and pallet. We discard that supernatant, and in a pallet we are going to add 100 microliter of protease buffer. Plus 10 microliter of 10% SDL plus 20 microliter of, sorry, not 20, 120. 120 microliter of 5 molar of NaCl plus 300 microliter of double distilled water plus 400 microliter of phenol and chloroform. This ratio is 4:1. See, when you get the pallet, in that pallet, you have to add a 100 microliter of the protease buffer, 10 microliter of the 10% SDL, 120 microliter of the 5 molar NaCl, 300 microliter of the double distilled water, and 400 microliter of the phenol and chloroform solution. Then again, we have to perform what? We have to perform a centrifuge. At 11,500 RPM for 10 minutes. Now you get a pallet, and so now in this you get a normal layer and the aqueous layer. Now you have to leave that normal layer and transfer that aqueous layer into the another tube. Transfer the aqueous layer into the other tube. And in that tube, you have to add a 1 ml of absolute alcohol. 1 ml absolute alcohol, and you have to incubate it at -20 degrees Celsius for 30 minutes. -20 degrees Celsius for the 30 minutes. Then you have to perform centrifuge at 12,000 RPM for 10 minutes. Then you get what? You get the supernatant and palette. You have to discard that supernet and into the palette you have to add 100 microliter of 70 sorry 100 microliter of 70% of ethanol. Then you have to perform a centrifuge at 10,000 RPM for 3 minutes. Then you get what? You get supernet and talent. You have to discard that supernet and talent. You have to air dry. Why we air dry the talent? To remove the ethanol smell. When it is air dry, we add 50 microliter of millipore water and leave it for the overnight. Leave it for the overnight at refrigerated condition. Then we perform egg roll. Here, procedure is clear. Procedure is clear. Please let me know in a live chat. Procedure is clear, then I will show you the experimental video. This video will also upload it to your account. You can see it, but here I will show you that video. Just here, please let me know in a live chat. And if you have any doubt, just let me know. Again, it is the same purpose. We have to purify our DNA. That's why we have to do our centrifuge at a multiple times so that the impurities will be removed. Here, Dimple, double distilled water is simply those types of the distilled water which are distilled by the two times. Okay. We all know that distilled water is what? Distilled water is the water which we get after the distillation process. Okay. So double distilled water means that the water which we obtain after the two times of the distillation process, that is known as a double distilled water. Clear to me? Clear to me? Now I will show you the experimental video of this. You see the video and let me know if you have any doubt.
Hello everyone, welcome to the ITLS Academy. Today we are going to perform isolation of DNA from human blood sample. But before start this experiment, we need few buffer solutions. The very first is lysis buffer, second is protease buffer, third is 10% SDS solution, and fourth is 5 molar of NaCl solution.
Also uploaded in your account with here I will show you. Okay.
So first of all, we are going to talk about how we are going to.
Some document is equal due to the recording. Okay. So just see the video for your explanation, and after that I will upload this video in your account, so that will be very easy for you.
Prepare lysis buffer. And what is the function of lysis buffer? Before I told you about that, what is the composition of lysis buffer, let me tell you what is the function of lysis buffer. Lysis buffer basically used for the disruption of cell membrane. Now we are talking about the lysis, what is the composition of lysis buffer and how we go. So the composition of lysis buffer is basically sucrose, tritone S100, magnesium chloride, and rich buffer. We require 10.96 gram of sucrose and 1 ml of tritone X100, 100 milligram of magnesium chloride, and 140 milligram of tris base. So this composition is required for the preparation of lysis buffer in 100 ml. But the percentage which I told you, we need to dissolve all these in 50 ml, and we need to maintain the pH 7.5, and after maintaining the pH, remaining 50 ml of distilled water we will add in the mixture. And now this is the 100 ml prepared lysis buffer. So this is our very first buffer solution which is used during the isolation of DNA from human blood sample. Now, second buffer solution which is required, that is protease buffer. Now, first of all, what is the function of protease buffer and why we use it in DNA isolation? So basically, protease buffer is used for the disruption of protein present in the human blood sample. The composition required for the preparation of protease buffer is sodium chloride, EDTA, and distilled water. We prepared this protease buffer solution in 50 ml, and we divide this 50 ml into 25 ml and 25 ml. First of all, we dissolve 1.25 gram of NaCl, 2.24 gram of EDTA, and in 25 ml of distilled water, and maintain the pH 8. And after maintaining the pH, we dissolve the remaining 25 ml of the distilled water in the solution. And now this is our 50 ml of protease buffer solution. Go. So next solution required is 10% SDS. SDS is basically sodium dodecyl sulfate. So we prepared it in 100 ml of distilled water, and for preparing this we required 10 gram of SDS and dissolve it in 100 ml. So the our 10% of SDS solution is prepared. So next solution is 5 molarity of sodium chloride solution. What we basically need to do, first of all, we need to use a formula to calculate how much weight we need to dissolve of NaCl in 100 ml of distilled water. And the formula which we used is weight is equals to number of moles into molecular weight of sodium chloride into 100 upon 1000. So our number of moles we are having 5 that is the number of moles. 5 molarity is what? Number of moles. And molecular weight of NaCl is 58.44 gram. And while we put these value in the formula, we get the weight 29.2 gram. So we need to dissolve 29.2 gram of NaCl in 100 ml of distilled water. And after dissolving, we get the 5 molarity of NaCl solution. So these are the 4 basic solutions which we required during the isolation of DNA from human blood sample. So now we are going to perform the isolation of DNA from human blood sample. For that, we already collect the blood from the laboratory. Now what I am going to do, I am going to pour 0.5 ml of a human blood sample in each of the Appin drop, and with the help of micropipette, as you can see, I pour 0.5 ml of blood in each of the Appin drop. And after adding this, we need to add 0.5 ml of lysis buffer in each of the Appin drop and mix it properly. So after pouring human blood sample in each of the Appin drop, now I am going to add 0.5 ml of lysis buffer in each of the Appin drop, and we need to mix them properly. And after pouring 0.5 ml of lysis buffer in each of the pen drop and mixing it properly, we need to centrifuge it at 11,500 rpm for 5 minutes. So after performing the centrifugation, now we are going to discard the supernatant. See, as you can see, here two layers are visible. This is a light color layer and this is the dark color layer. We need to discard this dark color layer. Okay, and after discarding the supernatant, we need to add 400 microliter of lysis buffer to each of the pen drop and repeat the process till we get the white pellets. And the centrifugation time and the rpm we need to maintain the same, 11,500 rpm for 5 minutes. After discarding the supernatant, I am going to add 400 microliter of lysis buffer to each of the pen drop. As you can see, I already set on micropipette 400 microliter. Now I'm going to add this much amount of lysis buffer to each of the pen drop and then centrifuge again at 11,500 rpm for 5 minutes. And we need to repeat this procedure till we get the white pellets. But you need to make sure that while you discard the supernatant, pellets should not be destroyed, and repeat this process till we get the white pellets. So after performing centrifugation for at least 3 times, we are having now the pellets. As you can see in the pen drop, we are having these pellets and we discard the supernatant very carefully. Now what I am going to do, we need to add 400 microliter of distilled water in each of the pen drop and mix it with pellets very carefully. We need to dissolve the pellets properly in the distilled water and then we need to perform the centrifugation at 11,500 rpm for at least 5.5 minutes. And after that we need to discard again the supernatant. So after adding 400 microliter of distilled water in each of the pen drop, now I'm going to mix it with pellets, as you can see. And after mixing, we need to perform the centrifugation again at 11,500 rpm for at least 5.5 minutes. After performing the centrifugation by mixing the 400 microliter of distilled water, now I'm going to discard the supernatant. Again, we need to discard the supernatant very carefully so that the pellets should not be destroyed. Now after discarding the supernatant of each pen drop, we need to add 100 microliter of protease buffer, 10 microliter of 10% HDA solution, 120 microliter of NaCl solution, 300 microliter of distilled water, and this is 400 microliter of mixture of chloroform and phenol. In this, for the preparation of this, we add 4 ml of phenol and 1 ml of chloroform. So now I am going to add 100 ml of 100 microliter of protease buffer. 100 microliter. See, now what we are going to do, we have to add 100 microliter of protease buffer. And now we need to add 10 microliter of 10% HDA solution. And we need to add 120 microliter of NaCl 5 molarity NaCl solution. Now firstly mix it slowly, and after mixing it we need to add 300 microliter of distilled water and 400 microliter of mixture of chloroform and phenol. So now I am going to add 300 microliter of Distill water to it. And now add 400 microliter of this mixture of chloroform and phenol. So after adding all these reagents, we need to again centrifuge it at 11,500 rpm for 10 minutes. So after performing the centrifugation by mixing all these reagents, you can see in the Appendrov that we saw two of the layers. The very first layer, second layer, and here is the pallets, as you can see. Now what we are going to do, we need to separate the upper layer, or we can say discard this upper layer. Discard it very carefully so that the other layer should not. And after discarding by the same manner, we are going to add 1 ml of absolute alcohol. As you can see, I already set that is 1 ml in the micropipette. Now after adding absolute alcohol to each of the Appendrop, we need to incubate it for 30 minutes at minus 20 degrees Celsius. So after incubation at minus 20 degrees Celsius, now we centrifuge the Appendrop at 12,000 rpm for 10 minutes. Now after centrifugation, as you saw, the white pellets are observed in the Appendrop. And we need to discard the supernatant again, and after discarding the supernatant, we need to add 100 microliter of ethanol to it and again centrifuge. So after discarding the supernatant, first of all, we need to discard it very carefully so that the pellets should not be discarded. Now after discarding the supernatant, we need to add 70% of ethanol of 100 should fly off, and after that we need to add tris buffer.
So this is all about the experiment. Do you understand? Anybody have any doubt related to the isolation of the DNA from from a human blood? Anybody have any doubt? Please let me know in the live chat. If everything is clear, just tell me. Let me know. Everything is clear. Okay, tomorrow we will discuss about the cell electrophoresis. So because every time I told you that after creation of the DNA, we have to perform an agarose electrophoresis, then we have to know what is this agarose electrophoresis. After discussing about the agarose electrophoresis, we will discuss the isolation of the DNA from bacterial cell, hair, and saliva. This will be discussed after tomorrow. Clear my point? Then on the last day, that is on Saturday, we will no. It is, give me 1 minute. So on 29, we will discuss about the PCR. Then on 30, we will discuss about the bioinstillation. Tomorrow, we will discuss about the agro's vessel. Okay. Clear my point? Should I end the class here? I am saying that tomorrow we will discuss about the agro's gel electrophoresis. Because every time I will tell you that I'm already told you that after isolation of the DNA, we have to perform a gross gel electrophoresis. So you have to know that what is the exactly a gross gel electrophoresis are. Then after that, we will move to the isolation part, that is your isolation of the isolation of the DNA from the bacterial cell, real and saline. Now it is clear, Dimple? Dimple, now it is clear from your side. Uh, we will let you know. Okay. Maybe it is on YouTube or maybe it is on. We will tell you tomorrow about it. Clear my point? Okay, thank you so much. We will meet on tomorrow.